part 2 of your Crash Course 9. Alright? Recall from our part 1 video, we were already done with your overview, particularly in DNA organization in your prokaryotic and eukaryotic cells. And secondly, we also discussed DNA replications. Di ba? Very easy process naman yan, sir. That's right. So class, for this video, I'm going to focus on transcription, translation, and their post-process modifications, and finally our mutations. Okay? So class, let's start our discussion on transcription. Alright, so class, remember from our discussion and our overview, I have said that transcription is the conversion of DNA to your RNA. And class, this actually serves as the first step of our gene expression or in gene expression. So what is gene expression? So that's just actually the transfer of our genetic information to our gene product. So it can either be, again, your different types of RNA or your functionally active proteins. All right, so class, here is the overview of our transcription process. Remember, from our overview of central dogma, sa prokaryotic cells, cytosol, because it has no sure nucleus, thus its location is in your cytosol. And on the other hand, for eukaryotic cell, ayan, remember, it's still inside the nucleus. So class, your replication is actually can either be a preparation for cell division or a preparation step for the transcription process. Okay, so always remember here as well that the transcription process only occur in your coding regions. Okay, when we say your coding regions, so these are the certain DNA regions that can code or produce proteins. All right, so these regions can produce mRNA, tRNA, rRNA. So class, these are the three types of your RNA that we will encounter in our translation process. But from our discussion of your nucleic acids, please also remember the other types of your RNA. Okay, so going back, your non-coding regions, on the other hand, ayan, they do not code for your gene products. And here, similar with our directionalities ng replication and the structure of DNA, always 5' to 3' ang direction ng synthesis. And here, class, very important to remember, the transcription process is actually the major point of our gene expression. Okay? So again, this is our major point of our gene expression control. So class, synonymous lang yan sa committed step in our gene expression. Because upon production ng ating RNA products, of course, translation will immediately occur. Okay? Your translation complex or translation assembly will then proceed upon formation ng ating different RNA products. And outcome here again, your single-stranded RNA transcript, particular lang yan sa mRNA. And then of course, we have your tRNA and rRNA. But class, remember here, we have an additional step to protect the integrity or the structural integrity of our RNA. Remember, because of its single-stranded structure, it is prone to attack. Okay? So class, that's why here, with the initiation as the starting point of transcription, elongation, and then termination, here we have your post-transcriptional processing. Okay? So this is your PTP class. So this is a very important process for your RNA to reach its mature structure. Again, we need to protect its structure against reactive products in the cell. Okay, so class, again, for the additional information on your transcription process, this is just a comparison or the diagram comparing the transcription for your prokaryotes and eukaryotes. So class, spot the difference between these two different transcription processes. See, almost similar. They use your RNA polymerase. They also have promoter regions where your RNA polymerase will bind. They also have your mRNA transcript that will be produced in your elongation step, and the termination will produce your mRNA for both. But here in your eukaryotic cell, we need your PTP or PTM, the post-transcriptional modifications. So we will discuss this specific step later on in our discussion. And class, additionally, very important to note here then, recall, in our DNA replication, we need your primer catalyzed by your RNA primase. Again, we added your primer initially in order to start the activity of your DNA polymerase. But here, in our transcription, there is no primer needed. So diretso or direct ang activity ni RNA polymerase in adding your nucleotides to form our initial mRNA transcript. Alright? So class, let's now discuss the specific processes here in our transcription process, starting with your initiation reaction. Well, let's start muna with your prokaryotic cell because relatively much is your initiation step for your prokaryotic cell. So again, the start of your initiation step is through the formation of your initiation complex via the binding of your RNA polymerase in your promoter sequence. Okay, so of course, this is with the help of your different enzymes and proteins. So class, for your prokaryotic and your eukaryotic cells, they have different promoter regions. All right, so here, ayan class, is a specific promoter region at -35. We have your TT-GACA. No need to memorize that. Okay, so for our second promoter region, it's located -10 region, your Pribnobax, the Tata at back. So class, just remember the Pribnobax. So both of them, they are located in your upstream site or upstream of your transcription start site. Sir, what do you mean by upstream? Later on, we will differentiate your upstream site and downstream site in your transcription process. So class, again, specifically, the RNA polymerase in your promoter will bind to it via the help of your sigma factor. So synonymous yan sa transcription factors in your eukaryotic cell. Again, for prokaryotic cell, ang initiation complex natin is composed of your active form. O diba, recall our term holoenzyme. That's the active form of your RNA polymerase plus its protein, your sigma factor in the promoter region. Okay, so that constitutes our initiation complex for your prokaryotic cell. And for regulatory sequences, of course, there are several promoters nearby our main promoter or our initiation complex. Okay, so before we proceed with your eukaryotic initiation, let's just briefly discuss the difference between upstream and downstream. Class, here is the start of our transcription. So this is the point of your RNA polymerase where it will start adding your nucleotides. So we also label it as your +1 site or your transcription initiation site. Okay, so ang upstream site is where or before the gene starts are being copied. So usually class or simply that's just before your transcription initiation site. So usual sequence or properties that you can see here is the promoters. Okay, so again, your promoters are always upstream your transcription initiation site, and your enhancer as well as your terminators are always downstream or after your transcription initiation site. Okay, so class, remember, upstream promoters, downstream enhancers, and terminators. All right, so that's the simple difference between upstream and downstream here in our transcription process. So let's go back with our discussion on the initiation in eukaryotic cell naman. O, diba? Much more complex. You can see here, of course, meron pa rin tayong promoter region. Here naman, class, ang very known promoter natin is your tata box. Recall, for our prokaryotic cell, box. For your eukaryotes, we have the negative 75 region, the kaat box, and the negative 25 region, the tata box. So ayan siya, class. That's just actually nearby your transcription initiation site. So this is your promoter, again, upstream your transcription initiation site. So class, di ba sa prokaryotic cell, your RNA pol binding needs sigma factor. For your eukaryotes, you have your transcription factors. But don't worry, you do not need to memorize these different transcription factors. Kasi class, this is very specific. We have your transcription initiation factors, transcription elongation factors, and your termination factors. But always remember lang that here in eukaryotic cells, ang counterpart ng sigma factor in your prokaryotes are your transcription factors in general. So for the initiation complex, so this is it. This is your initiation complex, class. So that's your transcription factor plus your RNA pol plus the promoter. Okay? So again, class, may nadagdag lang. So we just added your transcription factor here and your promoter as a part of our initiation complex. And here, a very unique for eukaryotic cells is the presence of your enhancers. Okay? So this will allow the binding of our different factors such as the elongation and other initiation factor. And here, remember, upon the start of transcription, you need to remodel your chromatin, diba? So we discussed the inactive and active form of our chromatin. That's why in order to start the transcription, we need to activate our chromatin. All right, so class, now we are done with the initiation. Very straightforward, comparing it to your replication process. So for the elongation, actually this is the rapid step. Basically, similar lang with the elongation step of your DNA replication. But here, instead of DNA polymerase, we will use RNA polymerase. So again, it will synthesize your RNA by adding your nucleotide, by reading the DNA template strand, and then unwinding the helix as it moves along. O, diba? Hindi na kailangan si helicase. Your RNA polymerase can do both. Diba? Slay. Of course, si RNA polymerase again has a polymerase activity and helicase-like activity. Let's check its different properties. Again, for elongation, RNA polymerase will move along your DNA template strand. O, class, emphasis on the template strand. So that's the strand that we only use in your transcription. So later, we will differentiate the coding and the template strand. And on the other hand, or additionally, it will also untwist or unwind 10 to 20 nucleotide bases at a time or in your RNA polymerase initiation site. Okay? So several RNA polymerases can actually transcribe the same genes simultaneously or at once. Ayan, for the speed, of course, last, no need to remember this. Again, very efficient ang addition ng nucleotides wherein your RNA pol can add 40 nucleotides per second. And remember, don't forget the sigma factor. This helps your RNA polymerase to accurately bind to its promoter. Okay? So here is the specific reaction occurring in your elongation. Class C, almost similar lang with your DNA replication elongation. But here, we are creating or producing your mRNA or in general, your RNA transcript from your template strand. Again, ang difference lang here, we are only using one strand, the template strand. Okay? So for our eukaryotes, ayan, your eukaryotic cells actually use three main RNA polymerase. All right? So of course, they are responsible, or each of them is responsible for transcribing specific types of RNA. Class, this is a typical structure of your RNA polymerase. Similar with DNA pol, it actually has many or numerous subunits. And similar as well with your DNA pol, we have three types. Here, class, ang very important lang to remember are their respective products and their location. Okay? So let's start. For RNA pol 1, it's located in your innermost location in the nucleus, the nucleolus. Okay? So RNA pol 2 and pol 3, that's just in your nucleoplasm. All right, for the abundance, of course, that's around 20 to 40,000 in our nucleus or inside the nucleus. That's why very quick or rapid ang elongation step because of the availability of this polymerases. And for the products, ayan, I would like to emphasize for pol 1, are responsible or mainly responsible for the synthesis of RR or ribosomal RNA, except the 5S subunit. For pol 2, class, generally, all of these are very important for the production ng mRNA. Remember, si hnRNA is your heterogeneous nuclear RNA, which is the unprocessed or newly made RNA. So of course, upon post-transcriptional modification, we will yield mRNA. O sir, anong relevance ni snRNA or your small nuclear RNA and microRNA? Class, they are participating in your gene expression. Or remember, the main control point ng gene expression is transcription. All right, for pol 3, again, main responsibility niya, production ng tRNA. And thus, again, we can conclude for pol 1, your RNA, for pol 2, si mRNA, and lastly, si pol 3, of course, don't forget tRNA. All right, so class, similar with our discussion on DNA pol, almost similar ang mechanism of the nucleotide addition. Okay? Si electronegative oxygen will attack the phosphate group of the incoming nucleotide and hence it will form a phosphodiester linkage. Again, our directionality is 5' to 3' in terms of the synthesis of our elongating chain. Clear? That's actually the end of our discussion on elongation. Class, now we are on your termination step ng transcription. And here, very short ang termination step for your prokaryotes and eukaryotes because similar lang ang ating mechanism with your DNA replication termination. So magdi-dissociate lang din ang ating transcription assembly. Okay? So here, the termination actually signals the end of your RNA synthesis with mechanisms differing between your prokaryotic and eukaryotic cells. Let's see the difference. For the signal, the main signal that your prokaryotes use is your DNA terminator sequence. Class, here in your terminator sequence, your raw factor will bind and hence, ayan, your RNA pol will be inhibited and thus it will detach to your DNA template strand immediately. Okay? O sir, pa-summarize, again, ang ating termination site is your specific terminator sequence where your RNA pol will bind or stop there with the rho factor. O diba recall, sa ating initiation, sigma factor ang need. In the termination, we have the rho factor. And after the binding, ayan, immediate po ang detachment ng RNA polymerase. So that's not the mechanism for your eukaryotic cell. Here, they use the polyadenylate or the polyadenylation signal. So very specific. That's your AAA or actually AAU sequence. So remember, diba, these bases are very weak because of its two hydrogen bonds. That's why that's very easy to cleave here in this site. So ang mechanism, of course, your RNA is cleaved 10 to 35 bases after your polyadenylation signal. And class here, hindi immediately mag-stop ang activity ng RNA pol. It will continue briefly before it eventually dissociates after cleavage. O diba? May buffer time pa siya before it completely dissociates in your DNA template strand. And of course, class, in your eukaryotic cell, our product is your premature RNA or pre-mRNA. Alright? So for your prokaryotes, ayan, they can directly use that messenger RNA for translation because they are tightly coupled, which we will see later. Ayan. So class, again, ang product natin in our transcription, of course, your single-stranded RNA. So this corresponds to your DNA template. O sir, medyo confused ako, ang daming terms. I know there's a DNA template strand, coding strand, sense strand, anti-sense strand, nonsense strand. O class, yung last wala yun ha. Again, we only have your sense and anti-sense strand. Okay? So here is the difference first between the two. So what we use in transcription is your template strand. Okay? And your non-template strand is the one that is discarded. Okay? So we also call it your coding strand. Sir, what's the reason? Because it is actually the similar with your RNA transcript. Okay? But here, instead of T, we replaced it with your EU nitrogenous or nucleotide. Okay? So class, here is the summary of your terminologies here. So your confusions are valid. So during my review, I'm also confused with these terminologies. But here, remember, si template is your antisense. Si coding is also your sense. So I actually memorize it as TACS or TACS. Template, antisense, coding, sense. Okay, for the description again, si template is the one used in your transcription, and this is read 3' to 5' because we synthesized it in the complementary direction, 5' to 3'. Again, this template strand is complementary to RNA, and the coding strand or your sense strand is actually, again, similar with our RNA transcript, but here, except your thymine is replaced by U. Okay, so class, again, the coding strand is not used by your RNA polymerase or not used at all in your transcription. Alright, so class, we are now on our last step ng transcription process with our post-transcriptional processing or your post-transcriptional modification. Class, here, we only have 5 main PTPs. So this include your 5' copying, splicing, and polyadenylation to produce your mature mRNA. Again, class, after the termination kasi, we only produce the pre-mRNA. So we need to prevent the degradation of that pre-mRNA via your PTP. Class, here is the summary or overview of our PTP. Of course, after transcription, immediately actually mag-occur ang 5' copying here. Okay? So afterwards, your splicing, cleavage, and polyadenylation will occur to form our mature mRNA. So class, here is the tabulated format of your PTP. First one with the 5' copying. So class, simply this is just a methylation reaction at the 5' end. So this occurs early actually again immediately after termination. Ayan class, again this is your methylguanosine functional group. So here we added your methyl to the guanosine nitrogenous base in order to protect your RNA, and of course it acts as a signal for ribosome binding. Here again, ang 5' copying is actually a methylation reaction only. Second, ayan, we have your polyadenylation. So from the name of the process itself, we will polyadenylate your pre-mRNA at its 3' end. So actually class, that's around 100 to 200 adenine added at your 3' end. So of course, for stability ng RNA and in order to protect it upon transfer from your nucleus to cytosol. Ay sir, lalabas siya ng cytosol? Yes, class, for translation, di ba? Remember from your undergrad biochem, your mRNA will exit the nucleus to participate in your RNA or your translation from RNA to your protein. Okay? So class, again, it uses your signal from the termination, of course, the AAU, AAA upstream of the cut site. So here is your signal. Ayan. And of course, after the signal is the addition of your 100 to 200 adenine nucleotide. Alright, so for our last PTP, we have your splicing. Familiar with the term introns and exons? Of course, your introns are the coding regions, while the exons are the non-coding regions. So imagine, diba, from your gene, the coding region ng ating genome, we actually have another intron and exon, or your coding and non-coding region. So class, here is your splicing mechanism. So this is actually occurring under or inside the spliceosome. So actually class, this completes your PTP to form the mature mRNA. And of course, we need again here splicing signals. Actually class, the splicing mechanism is a complex process, but we do not need to delve on the specifics of this process. Again, in the board naman, very surface level lang on the topic of splicing. Okay? So to enrich our discussion, of course, for splicing, again, this removes introns, your non-coding regions, and joins exons. So apologies class, napag-switch ko yata sila earlier. So again, ang introns are the non-coding regions, and your exons are the coding regions. So here, we will eventually form the mature functional mRNA. Alright, so class, again, this is the specific process of cutting the introns out of our mature mRNA. O see, your 5' cup and your poly A tail at your 5' and 3' end, respectively. But class, here, remember, ayan, we have an alternative splicing mechanism. So sir, what's that? Of course, this is just like a mix and match. So upon splicing ng introns natin, or to remove your introns, we are now left with your exons. And these exons can be mix and match. Okay? O check nyo. Here in our first protein, our exon 1, exon 3, and exon 5 were combined to form a different protein. Okay? Now, mix and match another, diba? So for protein 2, your exon 2, 3, and 4 were combined to form a new unique protein compared to protein 1. Okay? For our protein tree, ayan, exon 1, 2, and 5 naman ang minix and match. And here, your alternative splicing, is formally defined as your splicing that allows a single gene to produce multiple protein variants by joining exons in different combinations. Okay? And of course, even this alternative splicing occurs in your spliceosome. Again, your spliceosome is a complex of proteins, as you can see here, with your small nuclear RNAs or the SNRPs. Okay? So again, your SNRPs guide the activity of your spliceosome. All right, so that's it for mRNA. Of course, we have an additional post-transcriptional processing for your rRNA and tRNA. Okay, let's check. Ayan. For our ribosomes, of course, we have your ribozymes. So, di ba? I think very famous si ribozymes during our undergrad because it's very unique, di ba? It debunked the fact that all enzymes are proteins, di ba? So, very, not like the other girls, see ribozymes. So, again, si ribozymes are catalytic RNA molecules that act as non-protein enzyme. And class, very specific ang function ni ribozyme in PTMs. Of course, this is participating to form your ribosome or initial rRNA. Well, let's see. Again, for your ribozyme, this catalyzes the reactions like RNA splicing and cleavage in your PTP. Okay? So, for the significance, again, it disproved or debunked the idea that only proteins can be enzymes. So that's not true actually, class, because your RNA in its key properties can fold similar to your proteins because of their single-stranded nature. Okay? So another reason is that there are some bases of your RNA or a part of your RNA that have reactive functional groups that thereby induce folding. And aside from that, they can also form H bonds with nucleic acids. Diba? For instance, we have your self-splicing introns, and this one, class, is your RNase-P. Okay? A type of ribozyme that participate in RNA splicing. All right. So class, this is your ribozymes. Okay, so class, again, so for our rRNA and tRNA, very different ang PTP unlike your mRNA. So again, here for the two RNAs, they mainly involve cleavage, modification, and folding, diba? Rather than your splicing and copying that occurs or happens in your mRNA post-transcriptional processing. Or for the details, class, ayan, for ribosomal RNA, again, similar lang naman that we have cleavage from your large precursor, RNA transcript, and base modification. See, again, this ribosomal RNA forms the ribosomal subunits that will participate in our translation. Diba, recall the site of protein synthesis is in your ribosome. And here, there's no cupping or polyadenylate or polyadenylation occurring for ribosomal RNA PTP. On the other hand, ayan, class, before I forget pala, ayan, for an example of rRNA PTP, we have your 16S RNA. Of course, class, this structure has been produced during your PTP. All right, for transfer RNA naman, we have also your cleavage and then base modification and then somehow just a short intron removal. Let's check. Here it said that it forms cloverleaf structure. Class, again, this is the mature tRNA, and as you can see, really, that its structure is like a clover leaf. Okay, so in our discussion on translation, don't worry, we will also discuss the parts of your tRNA because again, similar with rRNA, this molecule participates in translation as carrier of amino acid or specific amino acids. So class, again, here of course, we will first do your cleavage, base modification, and the CCA addition. So class, again, this is your specific sequence that is added in your 3' end ng CCA. So sir, what's the reason? Class, that's the binding site of your amino acid for the translation process. Okay, so here, class, again, we will cleave this part, we will also modify this part in order to form this intermediate. And afterwards, after splicing as well, we will generate this mature tRNA. And later on, we will see that actually this tree sequence of DNA is what we call your anticodon for tRNA. Familiar with the term? Yes, sir, very familiar. All right, so class, we are already done with our discussion on transcription. Let's summarize the happenings, diba? Ang mga chika and happenings on transcription. Class, first, again, very different ang location, cytoplasm for the prokaryotes, and then sa eukaryotes, nucleus. Okay? Again, for RNA polymerase, we only have one single type for the prokaryotes and 3 types for your eukaryotes. So, diba, remember, Pol 1, rRNA, Pol 2, mRNA production, and Pol 3, mainly tRNA. For the initiation, sa prokaryotes, we have your sigma factor. Okay? This binds with your promoter, your cryptobox, diba? For eukaryotic cell, ayan, your TS or your transcription factors and your tatabox promoter. Class, please take some time to memorize the two different promoters. Si tatabox for eukaryotes and si cryptobox for your prokaryotes. Okay? Ayan. So class, again, for the promoters, very important in order to produce or to assemble our transcription initiation complex. All right. For the termination, diba? Sa prokaryotes, if we have your sigma factor in initiation, very easy to remember. Sa termination, we have your rho factor that will bind in your RNA pol at your termination sequence or termination or sequence. Okay? So it can either be raw dependent or intrinsic. Class, when we say intrinsic, meaning your RNA polymerase stops on its own without the need of extra or your support proteins. All right, for our eukaryotes naman, much more complex, but it often involves your poly-A signal or the polyadenylation signal. Diba, remember that AAU, triple A. Ayan, for RNA processing, of course, prokaryotes, direct to translation because it is coupled with translation. On the other hand, sa eukaryotes, we have 3 main PTPs, copying, polyadenylation, and splicing or your division. Here, of course, it is not coupled with translation. Remember, your transcription is an independent process. All right, so class, that's it for our transcription. Ayan, so class, we need to move on na, di ba? So we need to move forward to our next topic for this central dogma or CC9 discussion. And actually, this is the last process of your central dogma. And I just wanted to share, this is actually my favorite process among the three here in your central dogma. O sir, bakit? Because it's like a game, di ba? It's just like a puzzle that we need to decode from your mRNA to build a polypeptide. Naku, sir, ang nerd mo naman. Well, class, the decoding part is actually the most or the fun, di ba? The fun part here in your translation. So I hope similar din siya sa inyo. But before we proceed on that topic, ayan, let's have a brief overview of your translation process. Again, similar with your replication and transcription, the direction is 5' to 3'. But here, in terms of the way your mRNA is read, okay? For the location, as we have already reiterated, translation occurs in the cytosol and in particular in the ribosome. All right, for the outcome, again, your synthesized polypeptide chain will undergo post-translational modification. O parang similar siya with your PTP on your transcription process in order to produce your fully functional protein. Okay? So class, again, similar lang ang processes here in translation. With your initiation, elongation, and termination as well as TTM. So actually, class, even the factors are also here or present in your translation. But of course, very specific lang sila in your translation process. O see, that's our translation assembly. As you can see, we have your tRNA. O sir, nasan dyan? So class, this is your tRNA molecule. This is your ribosome, the large subunit, and your small subunit ng ribosome. And here is your mRNA. In mRNA, we have your codon being paired to your anticodons in the tRNA. Diba? So yeah. Oh, sir, may pag-game ka? No class. So that's just your genetic code, di ba? So I think you're familiar with this one. So this defines how mRNA codons correspond to amino acids, guiding accurate protein or polypeptide synthesis across nearly all organisms. Okay? So class, this is actually the fun part of your translation. The genetic code or the decoding part. But of course, in the boards, actually mga 1 to 2 items lang ang nag-appear about your whole translation process. Okay. So class, ayan ang tabular format of our genetic code. Oh sir, how do we use this? I think you're familiar. That these are your nucleotides and these are the first base in our codon. Remember, our codon has 3 RNA, 3 consecutive in particular RNA bases. And this one, so this is your first base in a codon. Similar with this one and this one. So class, you will check it here in our first row. Okay? So for our second base, here it is in our header of each column. Okay? Ang second base class is this one, and also this one. And thirdly, our last base or the third base in the codon is here in our last rows. All right? So class, here is your third base, your last base or your third base. Clear? Ayan. And of course, may mga features, di ba? Parang nagbebenta eh. May features ang genetic code natin. So class, remember, the key features of your genetic code. It has a codon system, non-overlapping and directional in terms of the codons in your mRNA. Redundancy or degeneracy, sir, what does it mean? Meaning, there are several codons that can code for similar or single amino acid. I will give you an example. Here, for instance, see UUU codon and UUC codon, they both code for your phenylalanine amino acid. So similar with leucine, we have two codons for leucine. For serine, ang dami, and proline, of course, we also have your four, diba? Four codons. Actually, class, kay leucine may additional four codons po nga. That's your redundancy or degeneracy, alright? So it's connected with our wobble hypothesis feature. So that's just your flexibility in the last base. Meaning, even though mag-change ang last base ng ating codon, it will still code to your similar amino acid. And actually, guys, that's a very strategic, that's the strategy that our genetic code used in order to prevent detrimental effects of your mutation. Okay? So again, wobble hypothesis allows us to code your similar amino acid even the third base is different. Alright, so correct reading frame meaning from 5' to 3' reading frame. And for universality, again, the genetic code can be applied across all organisms. So, diba? That's why it's universal. Alright, so class, to summarize again your key features, you have your codon system that we discussed. Non-overlapping and directional, again, from 5' to 3' ang direction. Read one at a time by our ribosomes. Thirdly, the redundancy or degeneracy, multiple codons can code for same amino acid, which is related to your wobble hypothesis, where the third base in a codon can vary without changing the amino acid synthesized. And of course, your reading frame and the universality. Okay, so class, please take some time to absorb the key features of our genetic code. Okay, so class, ayan, to brush up your knowledge or to test your knowledge on the genetic code, let's have some sample problems on the decoding, diba? So use the standard genetic code table to determine the AA or amino acid sequence corresponding to the following mRNA codons. So let's see, number 1, AUG. Sir, nasan yon? Actually, that's your start codon. Always see methionine ang start codon of our polypeptide chain undergoing translation. Second, GGC, that's just your glycine, diba? Here it is, class. And lastly, CAA, so that codes for your glutamine. Alright, so class, very easy, diba? Sana lahat ng board exam items, that's just like your decoding problem. Second item, UUU, so let's check. Phenylalanine. CCC, so that's just your proline. And lastly, GAG, diba? GAG, of course, that's specific for your glutamate. Alright, so very easy, class. And lastly, number 3, ACG. So si ACG is specific for your threonine. Si AAG is for your lysine. And lastly, UAA is for our stop codon. Actually, your stop codon, we have 3, diba? So si UAG, UGA, UAA, diba? I actually sing it like that in order for me to memorize it easily. Again, si UAG, UGA, UAA. Ayan, for your stop codon. Alright, so class, that's your genetic code. For translation, again, remember, maraming requirements, diba? Parang board exam preparation, you need to prepare the requirements in order to continue with your translation process. So specific requirements, we have your mRNA, tRNA, ribosomes, and your specific supporting enzyme, as well as your translation regulatory factor. So let's see. Ayan, so class, this is your, again, your translation assembly, including your large subunit ng ribosome and its small subunit. Here is your mRNA with its 3 nucleotides that we call your codon. And in your tRNA, we have your specific anticodon, which will bind with your codon via your Watson-Crick base pairing, diba? And your tRNA serves as your delivery molecule in order to deliver your amino acid to your site ng protein synthesis. Class, again, the codon and anticodon pairing allows accuracy. Alright? So class, let's check. Again, si mRNA, it carries the codon sequence that dictates, the amino acid order. Diba? Again, this is read from 5' to 3' direction. tRNA, of course, it carries your amino acid. We call your tRNA carrying the amino acid as your charged tRNA. Alright, so your anticodon from your tRNA pairs with your mRNA codon. And of course, remember, the site of protein synthesis is your ribosome, where it helps in aligning your mRNA and tRNAs to catalyze the formation of peptide bonds. Again, ang stabilizing bond in your amino acid chain, the primary of your protein is your peptide bond. And of course, we have several translation factors, particularly initiation, elongation, and release factors that regulate and assist each translation stage. Okay, so let's proceed with the specifics of your requirements here in translation, starting with the activation of your tRNA molecule. Again, class, the tRNA, we need to charge it with your specific amino acid. So here is the reaction or the chemical equation. So again, class, your preparatory step before translation is that the tRNA must be activated using energy by attaching or charging it with the correct amino acid. Here, always remember your enzyme that will catalyze this charging activation reaction. The amino acyl tRNA synthetase. Here is your specific reaction. The amino acid will be activated through your energy input in the form of ATP. And here is our intermediate, the amino acid with the condensation with your AMP, liberating your pyrophosphate molecule. And this intermediate will then be charged to your tRNA to transfer your amino acid to your tRNA and thereby liberating or displacing AMP. Plus, again, your charged amino acid, that's the terminology with your tRNA having an amino acid in its 3' CCA end. Alright, so class, again, very specific ang ating tRNA anticodon and mRNA codon base pairing. For instance, we have your AAG 3' AAG to 5' anticodon. Again, dapat complementary si anticodon and si codon. And thus, si codon specific for this tRNA will be your 5' UUC to 3'. And of course, it codes for your phenylalanine amino acid. Class, again, this is the structure of your tRNA. And here is the attachment site of your amino acid in your 5' CCA 3'. And alright, so class, please remember this attachment site. And here is your mechanism of your charging or activation of your tRNA. Remember, the enzyme again is amino acyl tRNA synthetase. Okay. Ayan. Of course, done with the tRNA, we also need your ribosome. Again, this is the main site kasi of your protein synthesis that ensures accurate alignment of mRNA and tRNA. And of course, class, your ribosome actually has an enzyme that participates in peptide bond formation. So for prokaryotes and eukaryotes, they have different small and large ribosomal subunits, but no need to memorize it. So it's actually hard to remember. But for the sake of our discussion, let's check. For prokaryotes, we have your 70S large subunit and 50S large subunit. All right. For eukaryotes, we have your 80S and 60S large and small subunit respectively. Class, again, no need to memorize this different subunits of your ribosome. Ang important here are the components of your ribosome. In your large subunits, we actually have your docking sites, and it is made up of 3 different docking sites. C, A, P, E. Again, class, hindi siya EPA because this is the entry point of your tRNA molecule as well as the entry point of your mRNA. That's why sa A site o C, that's your entry site for amino acyl tRNA. O, diba? A site, entry site. Okay, for our next site, this is your P site. So literally, this is your site where your peptide chain grows. Because class, here is the site of your peptidyl transferase. So later on, we will see that specific enzyme catalyzing your peptide bond formation. And of course, last site is your E site. So literally, the exit site for discharged tRNA, meaning it already transferred the amino acid from its CCA end. Okay, and of course, class, again, the peptidyl transferase is an rRNA-based enzyme of the large subunit. So here it is class sa P site where it catalyzes the formation of your peptide bond. Because again, ang P site ang site of our peptide bond or the growing peptide bond elongation site. Okay, so class, here is the overview again of your translation. So as you can see here, we have your initiation. So formation lang yan ng initiation complex assembly. So later on, we will discuss the specifics of your initiation process. So actually, this is just the much complex process relative to your elongation and termination. So again, second one elongation, third is termination, and similar with your transcription, we have here your post-translational modifications or PTM. Already na. Okay, so class, if you are ready, so let's proceed with our first process, the initiation step. As you can see here, this is actually the regulated step of translation. Now, don't be confused, ha? For the general central dogma process, particularly the gene expression control, transcription is the regulatory point. Here, for the translation, our initiation step is the regulatory point, and actually, this is the slow step because it sets up fully your ribosomal complex along with your mRNA and tRNA. So literally, class, that's your initiation complex. So let's see the processes occurring in your initiation step. So here is the visualization. Let's check. The first one is the initiation complex assembly, where your mRNA first binds with your initiator tRNA. And of course, upon binding ni tRNA and mRNA, your small subunit will scan for the start codon in your mRNA to attach it to your binding site ng mRNA and small ribosomal subunit. O class, ano nga ang binding site? That's the A site or your entry point, diba? So after malocate, ayan, we have your IFs, your translation initiation factors will assist or help in positioning the components, and additionally, as you can see, upon binding ng large subunit, we will need energy. Of course, that's the responsibility of your IFs, your initiation factors. Okay, and last, of course, this is your initiation complex assembly where your large subunit of your ribosome will join your existing subunit or small subunit. Again, class, ang ating initiation complex is consist of, or ang components niya is your small subunit, large subunit, your tRNA bound to your mRNA. The codon and anticodon pairing. All right. So here, your initiation complex is now ready for elongation. So ribosome is now set to begin the polypeptide synthesis. And as you can see, class, upon scanning your start codon, it will be immediately transferred sa T site in order to, ayan, in order for your next tRNA, sorry, to be accommodated. Now let's check the next process. O, diba? See, one slide lang, ah. Initiation step. So similar with your initiation step, your elongation step is actually a very easy step in your translation. O, see, the most rapid step wherein we grow your existing polypeptide chain by adding amino acids one by one to your growing or existing polypeptide chain. Ayan. So it is made up of three different processes. First one, codon recognition. This is the codon recognition class when your mRNA and your ribosomal subunits are recognized that a tRNA is incoming. Of course, it will accommodate it in your A site where your amino acyl tRNA will bind to the A site with the help of the EFs. Class, in the emergency fund yan, that's your elongation factor. Of course, we need to have an input of energy in order to facilitate binding. Ayan, as you can see, it's already bound in your A site. And your P site, particularly the peptidyl transferase, will initiate an attack in order to form your peptide bond in the second step. Particularly here, class, the amino group in your amino acid in the A site will attack the P site peptide. Ayan. So of course, class, that's the peptide bond formation. Or recall, your peptide bond again is amide in nature. And lastly, as you can see, after the successful binding or peptide bond formation, as you can see here, we will now translocate your tRNAs. Your uncharged tRNA here in the P site will be translocated in your E site. We call it your ribosome shift. Again, the tRNA in the P site will move to the E site and your tRNA in the A site. Sir, yan po ba yung charged tRNA? Yes, as you can see, it currently carries your growing polypeptide chain. Again, it will move to your P site as you can see here, and then your discharged tRNA will exit via your E site. Sir, the cyclable base tRNA? Yes, class. Okay, so it will undergo another series of your activation. Mag-a-add ulit tayo ng amino acid on its CCA. And then of course, afterwards, the cycle will just repeat. Your ribosome will be ready for next amino acyl tRNA. Again, here, the cycle continues codon by codon until the stop codon is reached where your termination will occur. Again, translation ends in termination when a stop codon signals the release of your polypeptide. O, diba? And here, it occurs in 4-stepwise process. O, let's see. This is the diagram, class. Again, when your ribosome detected that there is a stop codon, again, si uag, uga, uag, it will then facilitate the entry of this stop codon to the A site. And ayan, to the rescue na ang ating release factors. Class, not termination factors, ha. This is your release factors. And of course, they are very specific to their binding site. Si RF1 will bind to the stop codons UAA and UAG, while si RF2 will bind to your UAA and UGA stop codon. O sir, anong ganap ni RF3? Of course, class, that's the finishing touches in our third process. When your RF1 and RF2 are already bound to their stop codon, depending what is the stop codon in your mRNA, your RF3 now will process or trigger the hydrolysis between the polypeptide and tRNA bonds. Okay, so class, here it is, diba? So your release factor will detect if your stop codon has been accepted or accommodated in your A site. So afterwards, ayan, immediate hydrolysis ng polypeptide and tRNA bond here in order to, ayan, release your discharged or uncharged tRNA. And here, class, the last step, total disassemble of your translation complex. So here, the completed polypeptide is released from the ribosome, your tRNA exits, and the ribosome and mRNA dissociate. Okay. So class, of course, your mRNA is not recyclable, but your ribosome can be used for another protein synthesis. So actually, your ribosome will then proceed to another site of protein synthesis. All right, so that's it for our termination process for the PTM. Ayan, so of course, hindi pa tayo tapos. After your V polypeptide has been liberated in your termination step, of course, this will undergo PTMs to become fully functional and properly localized. So it's like you're going to activate, you're going to manipulate the structure of your existing polypeptide. O, di ba? C class, from your site of your protein synthesis, we have actually a signal peptide that will be recognized by our SRP. O, sir, sino naman yung main character na yan? Si signal recognition particle will actually bind to your ER. Ayan, sa ER membrane where there is a receptor protein. Plus, ang site ng post-translational modifications kasi is in your endoplasmic reticulum. That's why we need your SRP in order to deliver your whole protein synthesis site pa nga eh. And here, upon binding with your ER membrane, your ribosome will then be disassembled. And here you can see, ayan, the signal peptide is cleaved, and here you will deliver the protein or your premature protein. Okay, so class, again, we have different post-translational modification mechanisms. First one, the cleavage. So very straightforward, diba? Recall in our discussion on enzymes, we have the inactive form of your enzymes, your zymogen compounds. So of course, upon removal of your non-coding peptide segments here, such as your chymotrypsinogen zymogen, we will produce your active form chymotrypsin. Okay, so another strategy, amino acid side chain modification. So your R group of your amino acid can be acetylated, phosphorylated, and methylated. So basically, we are activating their side chain for spontaneous protein folding. Okay, so again, class, when we say activation, we just make our amino acid side chains reactive. All right. Thirdly, ayan, integration lang yan, class. Macromolecule coupling. Sorry. So again, this just produces your biomolecules, integrated biomolecules in particular. For instance, glycoproteins, meaning proteins plus sugars, and your lipoproteins, diba? There is an interconnection between your translation and your lipid digestion. And lastly, si co-factors for the use in our metabolic pathways. So of course, very familiar with our fourth PTM, tertiary structure formation. So that's the spontaneous reaction or process for our protein to be functionally active. Diba? Remember, not necessary naman ang quaternary structure for your protein to be functionally active. And of course, if we have tertiary structure formation, we have the assembly of our quaternary structure. Ayan. So again, class, these are discussed from our discussion on proteins. And lastly, protein targeting is a strategy that uses the signal peptides to guide your proteins to ER or other destinations. O see, not exclusive CSRP with our endoplasmic reticulum. So class, the protein targeting is the mechanism I have presented on the previous slides. Okay, so class, again, here is the summary of your amino acid side chain modification. O, diba? So as you can see, very important that we modify the side chains of amino acid in order to induce the spontaneous protein folding or for other biological roles. All right, so to end our discussion on translation, I have a short did you know segment here. Ayan, so class, did you know that actually your cells can translate one mRNA transcript into many proteins simultaneously in our structure called polysomes. So, diba? So as you can see, your polysomes are actually just your multiple ribosomes. We also call it your polyribosomes. So they are bound to a single mRNA transcript strand like a beads on a string in your electron microscope image. So class, as you can see here, here are your ribosomes. They are like your beads, and your string is like your, or it's your mRNA. Okay, so class, again, this actually boosts your efficiency in your translation, allowing rapid protein production in both, okay, both prokaryotes and eukaryotes, all from just one mRNA transcript. All right, so class, to summarize our discussion on translation, just remember our key components here. So we have your mRNA template, check. The tRNA, particularly its anticodon plus its charge, the amino acid attached, and its CCA. And we also have your ribosome, which is the site of your protein synthesis. Remember, it is composed of two subunits. Of course, first nagbabinds a small subunit, and then your transcription initiation factor will recruit your large subunit. And class, please remember the enzymes. The peptidyl transferase, again, that's a part of your large ribosomal subunit in the site in particular. And your amino acyl tRNA synthesis, sir, nagka-amnesia ako. Class, that's the enzyme that catalyzes the activation of your tRNA. And remember our special features again, direction, 5' to 3', genetic code, code, triplet or your codon, non-overlapping ang reading frame, redundant, particularly related sa wobble hypothesis, and lastly, universal to majority of organisms. Alright, so to summarize our processes here again, class: initiation, assembly po ng initiation complex with your small ribosomal subunit binding to your mRNA and your initiator, tRNA. And of course, si small ribosomal subunit is also the one that stands for the AUG, start, codon, and lastly, after your elongation, initiation factor has found your AUG, your large subunit will be recruited. Okay, for elongation, again, you have your codon recognition at the A site or the entry site, peptide bond formation on the P site, and your translocation going to the E site. Last, remember, this process is aided or guided by your elongation factors, diba? And for termination, again, this is a simple process where when your stop codon reaches the A site, ayan, your RFs or release factors are to the rescue. Si RF1 and RF2 will bind to their specific stop codons, while si RF3 will induce or will signal hydrolysis ng tRNA and polypeptide chain peptide bond formation. Ayan. So this will lead to disassembly ng translation complex. Alright, for post-translation, again, we have your folding, modifications, including your cleavage and phosphorylation, which are the main modifications in our proteins. And of course, it is targeted to your proteins final destination. Okay, so class, that's it for our discussion on translation. So for our last topic in this video, the part 2 of your Crash Course 9, we will briefly discuss mutations. So sir, do these topics appear in the board exam? Yes po. Particularly if you're familiar with the disease sickle cell anemia. So that's one of the common examples of your mutations. All right. So let's discuss briefly your mutations. Again, these are changes in genetic material, and in particular, it occurs mainly or spontaneously during replication. So these are being affected by your mutagens. Again, class, your mutagens will induce the change in your genetic information. We have 3 different mutagens: physical, chemical, or biological. Let's check. Ayan. For your physical, we have your radiation and heat. Class radiation, I think you're familiar, di ba? Your ionizing radiation, like X-rays, they can actually induce mutation. But of course, wag matakot, di ba? For your X-ray tests in your clinical lab, of course, small dose of X-rays lang yun. So of course, the benefits outweigh the risk of mutation. For heat, of course, it causes the purination of the structure of our DNA that will lead to thymine dimers or the formation of your reactive oxygen species that will lead to damage on the structure of your DNA. Alright. So class, for the chemical, we have your base analogs. So when we say base analogs, they are like, chemical imposter because they can sneak into your DNA in place of our 4 real bases. And of course, they mess up the pairing rules, thereby causing mutations. Ayun. So for intercalating and alkylating agents, so class, familiar with your ethidium bromide in your biochem lab. So of course, we do it for staining, diba? But of course, it's a mutagen, a chemical mutagen. That's why it's now being inhibited or not allowed in your laboratories. And lastly, of course, metal ions can intercalate or bind directly to your DNA, or particularly nga class sa phosphate backbone. O sir, what is the consequence of this? Of course, hindi nila ita-target yung mismong replication. Hence, they will block the repair systems, the repair mechanisms of our replication process. All right, for last, biological, of course, the bacteria and virus is a given. For transposon, sir, what are those? So class, your transposons are actually found in your chromosome. So they are naturally occurring, and we call it also your jumping gene. So they are mobile DNA sequences that can move from one region in our chromatin or in our genome to another. And of course, when they move, they will mess up our DNA sequence. Okay? So class, again, remember, ang bacteria and viruses like your SARS-CoV-2 can disrupt our cell regulation through DNA damage. So class, ang mutations, we can classify it into different types. Your point or base pair mutations, so they are just the same, and your insertions and deletions based on how our DNA sequence is altered. All right. So to start discussing on the classification of your mutations, let's just watch this short video.
Gene mutations are changes in the base sequence of the DNA in a gene. They can be brought about in several ways. Assume that the normal base sequence on a coding strand is ATG, CAT, GTA, and TTG. The amino acid sequence produced should be methionine, histidine, valine, and leucine. If one or more bases are deleted from the coding strand, the reading frame will be shifted. Thus, the amino acid sequence after the point of mutation will be altered. The resulting protein is usually non-functional. Similarly, if one or more bases are inserted into the coding strand, the reading frame will also be shifted. And the amino acid sequence after the point of mutation will be altered. The resulting protein is usually non-functional too.
Class, for our replication process, usually base pair substitution ang most prominent. So let's focus our attention on this third type of mutation. Now let's check.
If a base on the coding strand is substituted with another base, a triplet code on the strand will be changed. This may result in one different amino acid in the polypeptide produced. If that amino acid alters the conformation of the protein sufficiently, the mutation would have an obvious effect on the phenotype.
So actually, that's that specific type of mutation on your sickle cell anemia. So don't worry, we will discuss that later. All right, for our last type of mutation.
Similarly, if the order of bases on a coding strand is changed, a different amino acid in the polypeptide may also result.
All right, so class, that's it for our intro on the classification of your mutations. Again, class, we have three main types. Point mutation, we have your insertion, and we have your deletion. First, we will discuss the subtypes of your point or base pair substitution. So this includes your silent, missense, and nonsense classification. So each of them can affect your protein production or expression differently based on how they change our DNA code. So again, class, ang point of our mutation is target is in your DNA replication. That's why from your DNA template strand to your mRNA transcript to your proteins, of course, it has a domino effect. All right, so let's check our first type, your silent point mutation. So clearly, from the name itself, silent in terms of its amino acid product. So it does not alter the amino acid or protein function. Again, our reason here is your genetic code redundancy. Very flexible po kasi ang last base or third base natin because of the wobble hypothesis. Okay, so let's check the specific example here. So class, here is your original translation. So I think it's also in your manual. So upon silent mutation or when your silent mutation occurs, here instead of your G, we replaced it by A. And of course, your template strand will have that E base instead of your what is the base pair of your A of your G, your C dapat, diba? But instead of C, as you can see here, it is replaced by uracil. And of course, because your GGC and GGU both code for your glycine, your mutation will lead to your silent point mutation. Okay? So sir, meaning harmless ang silent mutation, not necessarily harmless class. Because it can affect our gene expression without changing the protein. So they can inhibit or activate a certain gene nearby to it when an amino acid, I'm sorry, when a nucleotide sequence has been changed. Okay, so again, your silent mutation simply can affect gene expression by inhibiting or activating your nearby genes. Okay, so class, let's proceed with our next two types of your point mutation. Missense and nonsense mutation. Let's go to the first muna. Missense mutation meaning the substitution of one amino acid for another can occur and may alter protein structure and function. Let's see. As you can see here, again, this is the original translation. And here, instead of C in this place in your template strand, it was replaced by T. Diba dapat G yung nasa complementary strand niya, but here, it becomes letter A or your adenine. So class, of course, domino effect, sir. Yes. So A instead of G in your mRNA transcript. And as you can see, class, very heavy or very detrimental ang effect kapag nasa first base ang change. From your original na GGC, it became AGC, diba? So si GGC codes for glycine, but the AGC codes for serine. And class, of course, if the nature of your amino acid is really different from the new one, it will induce a conformational change in your protein folding. Diba? For instance here, glycine is actually a hydrophobic amino acid. While serine, it has a hydroxyl group in its side chain, so relatively, it's a hydrophilic amino acid. So of course, that shift to your hydrophobic to hydrophilic property will change as well the conformation of your protein. And of course, it will lead to a non-functional protein. All right. So on the other hand, kay nonsense mutation, we call it nonsense because the original amino acid codon was turned into a stopped codon. And of course, it produces a truncated, usually non-functional protein. See here, class, in our example here below, A instead of T in your the template strand, and thus in your mRNA transcript, it is U in your codon rather than A. And remember, CUAG actually codes for a stop codon. So this is a type of your nonsense mutation. A non-functional protein is obtained here in this type of point mutation. Let's proceed with our last batch ng mutation type. Primarily your insertion and deletion. So these two, basically this just adds or removes one or more nucleotide pairs into a gene. So let's check each specific mutations. First, when we say insertion, so very basic, sir, we will just add a nucleotide pair. Yes, here we have an extra A, and thus we will have an extra U in your mRNA transcript. See, of course, UAA still codes for a stop codon. So actually, class, this is also a non-functional protein. For deletion, of course, if in addition or insertion, we will add here, we will remove a nucleotide pair. So for instance, A is missing here, and thus in your mRNA transcript, U will be missing. See the domino effect here, and class, of course, it will give birth to different amino acids with different nature. Of course, it can be detrimental depending on the nature of the amino acid that has been changed. All right, so class, here is our did you know on your sickle cell anemia. Class, please focus your attention here, because again, this is a board exam question. So again, a single letter change in DNA can reshape an entire cell, literally, diba? From your original or your natural hemoglobin molecule, it was turned into a sickle cell shaped red blood cell. As you can see, a single point mutation can have life-changing consequences, particularly in sickle cell anemia. A mutation in the beta globin gene swaps just one amino acid. Single point mutation lang siya, class. And here, our result, glutamic acid, diba? This is a hydrophilic acidic acid, amino acid rather, is replaced by valine, a hydrophobic amino acid at position 6. Sir, ano pong result? Of course, this tiny change from its amino acid sequence makes hemoglobin clump or stick together, and thus distorting the red blood cells into its sickle shape. Ayan. So class, again, this can block blood flow kasi, and thus it will reduce the efficiency of your oxygen delivery ng hemoglobin. Of course, this causes red blood cells again to become sickle shape, of course, and making them less flexible and block blood flow. All right, so class, again, remember our important information here. Replaced your glutamic acid is replaced by valine at position 6. Okay. So class, we arrive now here at our Lars words, your favorite slide. Diba? So let's summarize our discussion on central dogma. Let's see. Again, for the direction, replication, copying of DNA. Transcription, it is the transfer of your genetic info to your gene products, DNA to RNA, and its different types. For translation, of course, very specific only to your mRNA to be transformed or converted via translation to your fully functional protein. So, ang location class, again, we will emphasize only your eukaryotic cell for replication and transcription. In order to protect its structural integrity, of course, it needs to be inside your nucleus. And of course, remember, the RNA will exit the nucleus to participate your translation process in the cytosol, particularly in your ribosome. All right, again, class, remember also our key biomolecules. So in replication, we encountered polymerase as our main elongation enzyme, helicase to unwind your original DNA double helix, and ligase specific for your lagging strand in order to seal the gaps or the nicks in between our Okazaki fragments. For transcription, of course, direct addition of your nucleotides. No need ng primers, diba? With your RNA polymerase as well as our transcription factors. And lastly, for translation, we have your ribosome, tRNA, and your enzyme that catalyzes the activation of tRNA, your amino acyl, tRNA synthetase. And of course, class, in terms of error correction, remember, majority of errors or mutations occur in your replication. That's why we have a lot of proofreading and much repair mechanisms here in order to minimize those mutations. For transcription and translation, we do not have actually that much repair mechanism. All right, and lastly, for mutation impact replication class, again, this is a domino effect, permanent ang impact, and this is actually heritable changes in our genome. That's why it serves also as a point of variation that will serve as an important role in evolution, di ba? For transcription, ayan, the misregulation ng gene expression through RNA synthesis is one of the heaviest or detrimental impacts ng mutation. And for translation, as we have seen, again, we have different types of mutation, and a majority of them can alter the protein sequence and their protein structure. All right, so class, again, this is a meme that summarizes our central dogma. Starting from our genetic info in the DNA, it teaches niya yung information to your RNA, our gene product, and finally to your bestie, si protein. All right, so class, this is the end of our discussion on Crash Course Night. Ang haba, no? So actually, by far, this is the longest crash course in your biochem subject. And with that, ayan, to wrap up our discussion, please answer your practice test 9 about central dogma. So it involves your replication, transcription, translation, and your mutation. So class, again, please practice answering closed manual PT answering. Ayan, so class, again, please take some breaks, ha? And of course, don't forget to